We will now listen to Mrs. Cristina Martín, and she will tell us about manufacturing of hope. What are the projected timelines? I think she comes from Viralgen. Is that correct?
Yes, that's
Thank you
correct.
very much. You're welcome.
Thank you.
Thank you.
Okay, so, well, first of all, I would like to thank you for inviting us to this conference. It's amazing the work that you have done and all the work behind all the foundations that are here today. So I'm going to talk about the manufacturing of a hope, the manufacturing of CTN and V1 product and the projected timelines. But I would like to start introducing myself. As he has mentioned, Gregor has mentioned, my name is Cristina. I was working for Viralgen for more than 3 years and a half, and I'm biotechnologist by training, and I am the project manager in charge of CTN and V1 program in Viralgen. Okay, so first of all, I would like to start with a question to make this more interactive. So please, can anybody from the audience let me know what can you see in the screen? Zeros, yes, we have a bunch of zeros here. In fact, we have 13 zeros. So what happens if we change the first zero by a 1? We are obtaining a very huge number, which is 1,000 billions. And for you to have an idea of the dimension of this number, we are talking about more than 1,000 times the people that are currently living on Earth, which is a lot. And probably you are wondering why I'm talking about maths and numbers when this is related to gene therapy. So it has a sense. This is the number that we need to produce by average for a patient to be treated with gene therapy. So this is the number of small vehicles. You have heard a lot about them during the previous talks. So these small vehicles, therapeutic vehicles, are the AAVs and contain the right piece of DNA, in this case, CTN and V1 gene, okay? So they are really tiny molecules, and we need to deliver this huge amount of molecules through the tissues to the cells that we need to bring the gene in. And in this case, these cells are part of the brain, okay? As, for example, Andrea has explained in her talk. So I would like to give you a brief introduction about our manufacturing process. So continuing with the parallelism of the vehicle, I would like to explain you our manufacturing process as a car, okay? So for that, we need the engineer. The engineer is the producer cell line, and we need to give the engineer the proper information to produce the AAVs, okay? So for that, we are using 3 different pieces of DNA that contain the information that we need to provide the cells to manufacture these AAVs. So we will use 3 different pieces of DNA. The first one is the packaging plasmid that contains information about the shell of the AAV, the outer part. So in this case, it will correspond, for example, to the number of doors that the car has, the windows, or the color of the car, okay? Then we have the transgene plasmid, that is the CTN and V1 expression cassette. That is the passenger that goes inside the car. So it contains the corrected gene and is delivered to the cells that we want to modify. And then we have the helper plasmid that is providing the tools that the engineers will need to build this car. So for example, the hammer, the screwdriver, etc. So we mix all these pieces of DNA, these pieces of information to generate a cocktail of DNA, and we introduce that inside the cells. So we call this process transfection, and we need to transfect more than 1 million cells per milliliter, which is a very high cell density. So we need some weeks to reach this cell density. So after the weekend, the cells are manufacturing our car, our vehicle, our AAV, and then we purify it and vial as in the format as you can see in the image, okay? So where we are doing all this process, so for that at Viralgen, we have 13 different GMP suites. I was going to explain what GMP means, but they have talked a lot about this term. So you can see in the image some of the bioreactors we are using on our installations. And these 13 GMP suites are located in 2 different buildings. The building you can see in the right side of the screen is the clinical facility. So here we will produce the different vectors that will provide to CTNNV1. And then if jumping into commercial, we have also the commercial facility in which we will manufacture the CTNNV1 product if moving to commercial, okay? So just to give you a bit of context about our company, it is a joint venture between ASBio and Columbus Venture Partners. So ASBio was providing the technology and the deep knowledge about AAVs and also about drug development, and Columbus was providing the funding. So it was raised in 2017, and in 2020, it was acquired by Bayer, giving the final boost to this project. So we are located in the north of Spain in a very beautiful city called San Sebastian. And now we are more than 400 employees. But as you can see in the next slide, we started just with 7 people. We call them the 7 Magnificent because they have done a very huge work. And along over these years, we have achieved different milestones, but I would like to stress out the GMP clinical certification for the clinical facility and also the commercial certification. And in 2021, we opened the commercial facility that I was showing you in the previous slide. So through all these years, and thanks to the huge effort of our team, we were able to manufacture more than 1,000 batches. Out of them, more than 100 are GMP batches, and some of them are enrolled in different clinical trials, specifically in 27. We are working with more than 65 clients, and we are not working just with companies. We are also working with foundations, because for us, the corporate social responsibility is really important. It's a key part of our DNA, and one of our main objectives as a company is to work with foundations to help broadening the access to patients to these gene therapeutic treatments. So we are working with different foundations, as you can see in the image. Probably you have heard about some of them. We are working with Columbus Children Foundation, with Curious PG50, Phoenix Nest, Elpida, Hannah So Foundation, among others, and now with CTNNV1. So going back to CTNNV1 project, what we are going to perform, well, we have already performed the first step. I will show you later the results we have obtained. But we have manufactured, as I was mentioning, a 2-liter scale. When I'm talking about scales and liters, I'm talking about the cell culture with which we are beginning with. And in this case, this feasibility scale has a double purpose. The first one is to ensure that the product is fit into our platform, because here we are not talking about a predefined recipe or predefined chemicals. We are talking about complex biological systems. So we need to ensure that the product of its client is fit into our platform, and we are able to provide a good productivity and a good quality product, okay? And then the other purpose of this scale is to provide enough material for research studies. Then we have the 50-liter scale that will be intended for toxicological studies, and the last scale, that is the GMP scale, will be intended for the clinical study. So going back to the manufacturing process, I would like to zoom in a bit on the 2-liter scale. So probably you remember this picture that I shown at the beginning of the presentation. As I mentioned, this is called transfection, and it's triple plasmid because we are using 3 different plasmids, 3 different pieces of DNA. So then we are letting the cells manufacture these AAVs over the weekend. And then we continue with the purification of these AAVs. Through the process that you can see in the screen, we have several steps to obtain the final product, because the aim of this purification is to remove all the impurities that we can have in the product. Then this final product is stored below -60 degrees. So here are the timelines for this manufacturing. So first of all, I would like to show you the timelines for the 2-liter manufacturing. So as I mentioned previously, this, sorry, the letters are moved, but as I showed you previously, we have already manufactured this 2-liter research rate run. We were also working on the transient plasmid amplification in an outsourced facility, and we were able to already manufacture the AAV. Now we are in the quality control step, in which we are doing all the assays that we need to perform to ensure the quality and having a good productivity for this product. So we will end them on next week, and we will be able to close all the documentation that will be provided to the CTN-NV1 Foundation. So here you can see as well the timelines projected for the 50-liter run and the GMP run. So these are tentative timelines, and just to let you know, as a rough estimation, we will start with the preclinical manufacturing, the 50-liter run, by the middle of Q3, ending this manufacturing run before the beginning of next year, and we will start in the beginning of next year with the GMP run. So as you can see, we will perform the manufacturing of the 50-liter, and then the QC characterization and revision. And as well for the GMP run, we will perform the QC characterization and revision. And this is taking a lot of time, because we need to ensure the quality of the product, and also to do all the documentation that my colleagues were talking about, which takes a lot of time. Okay. As you can see as well, we are outsourcing the transient plasmid amplification for both scales. So I wanted to show you some of the results we have obtained for this 2-liter run. So on the right side on the screen, you can see the results for ITR-ddPCR. So this is a measurement that allows us to know the concentration of vector genomes that we have, and is a measurement of the quantity of AAVs that we are having in our final product. So here you can see the average of Viralgen historical data for AAV9s. And you can see that CTN-NV1 product is performing well, since this is above the average of historical data that we have obtained. And also for the 2-liter scale run in the lower part, this is the ITR-ddPCR titer as well. CTN-NV1 product is in the 70th percentile. So the same for the percentage of full capsids. This is the capsid that contains the complete expression cassette. So you can see that CTN-NV1 product, that is rounded in red, is about our Viralgen's historical data, and it is in the 62nd percentile. So I cannot end up this presentation without saying thank you on behalf of the whole Viralgen team. As I mentioned previously, this is very important with us. We are very happy to work with foundations like yours. Here you cannot see the complete team, because this picture was taken 2 years ago. But for us, it's a great source of motivation to work with foundations like yours, and gives sense to the work that we are doing in a daily basis. So thank you all for your attention.